A warm welcome to the webinar Single Cell and Flow Cytometry Profiling of Tumor Microenvironment, jointly organized by Singleron and Cytech. This is Inting, and I will be your webinar host today. The tumor microenvironment plays a key role in tumor progression, treatment response, and disease outcome. Today, we're honored to have two speakers with us who will talk about two powerful tools for tumor microenvironment profiling on the single cell level. So first, Dr. Sujata will introduce single-cell sequencing and tissue dissociation, followed by Dr. Mark Weider, who will share some interesting data using flow cytometry and Singleron's tissue dissociation solutions. Dr. Sujata is a regional application manager at Singleron and an expert in genomics and single-cell analysis. So now let's invite Dr. Sujata to give her presentation. Sujata, please.
Good morning to everybody. Thank you very much for joining this webinar session. And I'd also like to thank Dr. Ma for being part of the webinar. I'll quickly start by introducing Singleron Biotechnologies and some background about us. So we are a company that focuses on single-cell solutions. We do kits, instruments, and software to support the single-cell analysis workflow. We are a global company present in Europe, US, China, and here in Singapore. And at all of these locations, we also have service labs. And in a minute, I will explain what that means. So just to start, we all kind of know why we need single-cell sequencing, but just to remind ourselves, if you do a bulk sequencing of a sample, you get an idea on the mixed cell population, a pooled result, and an average profile of all the different cell types in the sample. But if you're actually looking at the single cell level, you're able to precisely profile the different cell types in that sample, which is important for facilitating downstream processes like drug target discovery or biomarkers. So the topic of our webinar today, the tumor microenvironment, this is a sample that fits right in because it's important to understand this complex microenvironment since it's a very key factor in understanding tumor progression, the treatment for it, response to a treatment, and overall disease prognosis. And this is because this particular environment has a whole range of different cell types. You have the cancer cells, immune cells, stromal cells, blood vessels, and depending on the tumor, you have adipocytes, neurons, etc., and of course, non-tumor cells of the same cell types. And so it becomes obvious that single-cell technologies such as single-cell sequencing and flow cytometry both are important because they will help you not just capture these different cell types, it helps you understand the interaction between these cell types, cell dynamics, and their interplay. Okay, so since there is a good percentage that has not done single-cell workflow before, I will just start off by giving you the steps that usually are involved in such a workflow. You have to get your tissue and preserve it, dissociate the tissue, which is one of the parts that we will be talking about, and then partition into single cell, library prep for sequencing, and then finally data analysis. And we at Singleron have solutions for all of these steps, except sequencing. And I will go through the most important ones that will be relevant, especially if you're working with a tumor sample. I also want to point out here, as mentioned, we do services. So of course, you can get products from us to support each of these steps. But if you want to just give us the sample, we do end-to-end single-cell service at our service labs, including the one here in Singapore, where you can consult with us on the project and then just ship us the sample. And we will do all of these steps for you and give you back a data analysis or any customized bioinformatics that you would require. So coming back to our solutions, when you're starting off, say you have gotten a tumor biopsy and you're not ready to process it immediately, you need to preserve this tissue. We have a solution for this. This also helps if you need to ship this into our lab somewhere. And this solution will help preserve the tissue for about 3 days at 4 degrees with good viability. And that's sort of the data that I've shown here. And once you've preserved and ready to process your sample, the first step will be tissue dissociation. For any single cell workflow, you need to get your tissue dissociated into single cell suspension with good viability and good cell numbers. And that's where our solutions come in. So ours is an enzymatic plus mechanical dissociation setup. So for the mechanical part, we have our instruments and our unit. So as it sounds, it's a mechanical breakdown of your sample using this unit and the little blade structure in here, and together with a dissociation solution, which is the enzymatic part of our dissociation setup. And so what we are able to provide is very efficient dissociation using our system. So you get cell suspension with high viability and high yield. And the operating time of most of our programs is 15 minutes. And we also provide a heating at 37 degrees. We've tested a broad range of tissue types across species, so it's very versatile to use, and also tissue inputs depending on the size of the sample. If it's a small biopsy, as low as 10 mg to 4 grams of tissue. And finally, depending on your needs, you can go with an 8-channel high throughput option or a 2-channel independent option. And both of these have flexible programming depending on your tissue type, and you can go between 30 to 300 RPM in the physical homogenizing of the sample, and also set parameters such as clockwise, anti-clockwise looping of your machine. The use is very simple, just a workflow. You open the unit, add your sample, solution, run your program, and you're ready to take out your sample for downstream application. Of course, if you're doing single cell work, you may want to consider filtering, doing a QC, and any optional steps such as red cell removal or debris removal. But this dissociation system has varied applications. Single cell sequencing is what I will focus on in my webinar here, part of the webinar. Another very important application is flow cytometry, which is what Dr. Ma will be covering in the second part of this talk. And he will cover where they have actually processed using our dissociation system lung cancer samples to test and develop a 20-color flow cytometry panel. Other than this simple cell sorting or setting up a single cell culture, all downstream applications after using our dissociation system. So just some data to show that we've tested many different samples, and you can see that we get good viability and cell yield. And we also have publications that use our solution. So here, liver and brain metastases of brain cancer tissue was used from human samples. And here it's used on mouse testis samples to do dissociation of the sample. And like I mentioned, here's one publication where the tissue dissociation, our solutions were used not for single cell work, but more for flow cytometry experiments. And here it's a breast tissue sample. And so I want to just end by also mentioning, I know a lot of people I see in the poll did manual dissociation. So is there, why would you want to switch to or also consider doing automated dissociation? So we got some internal data that we use skin to carry out both manual and automated dissociation. Now, skin in general is a tough tissue to do dissociation. And what we find is using an automated option, you have improved cell yield, which means you will detect more cell types and subtypes that you could be missing in the manual. And there is no compromise to gene expression profiles if you used an automated. So it is good to consider an automated solution for your dissociation. So just quickly going down to the other steps, the main part after you have a single cell solution is to do cell partitioning into single cell and labeling them. And for this, I'll just bring up our core technology. We are a microwell-based technology, which means our cell partitioning is done in a chip like this, which has microwells. We have 3 versions of this chip, depending on the number of cells you want to capture and cell sizes. And the second part of our core technology is barcoding beads, which has the barcodes that would label your cell individually. Now you can choose your method of loading your cells, beads, and all other reagents into this chip. One is a manual option, by which I mean you can buy our kit. No specialized equipment needs to be purchased. You have a pipette and you can load all of this. And the advantage here is you can view your cell loading or bead loading under any standard microscope. Of course, if you want to step away or looking for a high throughput option, you have the Matrix Neo. Here you can run between 1 to 4 chips in one run. And also you can also just use the required channels, by which I mean you can also just do one sample at a time. And here you have the advantages of running the different chips, the different configurations we have together. So we are able to provide mRNA profiling, which is our GEXscope kit. So this will be what the bead structure looks like in that kit, but also multiple other multi-omic options. I'm just flashing here the main assays we provide. So we have the immunoprofiling assays. We have a kit that can do single nucleotide variations, gene fusion, and splice sites, and also non-poly RNA. For example, viral transcripts at the single cell level, some specialized unique solutions that do surface glycosylation level measuring and RNA dynamics, and finally a sample multiplexing kit. For a tumor microenvironment question and a tumor sample, these are probably the most relevant solutions. So I will just cover these quickly. Just starting off by showing that our kits have been used in various different sample types. Here's a snapshot of publications that are on human samples. These are on lung tissue, breast tissue, and also colon cancer tissues. And we just also have tested our kits on various different species, mouse models, Drosophila samples, and also non-human primates. You can also profile up to 30,000 cells using our solutions. And what that helps you do is pick up very rare cell types. So for a sample such as a tumor sample, and if you're interested in studying the tumor microenvironment, this is a good option. As you can see here on a whole embryo sample, we're able to pick up rare cell types like the T cells. And our solution is also able to pick up capture sensitive cell populations. So one example would be neutrophils. So these are, some of these immune cells can be very sensitive to cell capture methods and be lost in the data, whereas we are able to capture this. Here's a bone marrow sample from mouse, and you can see more than 40% of the sample are neutrophils. And here's a publication that we have where neutrophils were captured from the pancreas of mouse samples. And then the other thing you may want to do if you're working with a tumor, interested in the immune cells and the immune microenvironment, we have two kits to profile this. The first one is a VDJ-based kit. And here, if you're interested in the CDR3 region, for example, want to know the T cell and B cell clonotypes or the cell diversity, just monitoring the immune response, this would be a kit you can consider. So along with the transcriptome, you also get CDR3 regions of the T cells and B cells. But if your application is further down, you want to develop personalized immunotherapy. So you need to understand the whole T cell and see how it's specific to diseases or any antigens. Then you want to consider our full-length immunoprofiling kit. Here you will get both the transcriptome and the full-length immunoreceptor. So now we're at the end of the workflow. You have your library. Our products, our libraries can be sequenced on all established short and long read sequencing platforms. And we also have bioinformatics solutions to get you from the raw FASTQ files to all usual single cell analysis. And we also have a single cell database that you can leverage on if you're interested in clinical studies. So I won't go into detail into these, but these are all available for you to use. I will end off by saying that, just showing a customer publication here, which has looked at tumor microenvironment using our solutions. So here it's hypopharyngeal squamous cell carcinoma. So the sample was taken, the hypopharynx, and they have different tissue types, normal, primary, tumor, and the node. And first, they were able to use our single cell solution, JEXscope, to do these, understand the different cell types in the sample, the immune cells. And on top of that, they were able to add the TCR repertoire for all the T cells in the data. So here you can see they're able to understand the differences in the diversity, different clonotypes, what cells have TCRs on a UMAP, and finally get the top 10 clonotypes, which would be of interest for downstream questions. I'll just do a quick summary before ending. The take-home message is that we provide sample to solutions for single cell workflow, starting from tissue preservation all the way to data analysis. And all of this, you can also leverage on our end-to-end single cell analysis services at the single RON labs here in Singapore and in other places. And of course, any single cell requirements you have, please feel free to reach us at the website or also our sales email. And do follow us on our LinkedIn to keep updated on what's happening. Thank you.
Thanks so much, Sujata. We have time for maybe 2 or 3 questions. So everyone, please use the Q&A function if you would like to ask any question to the speakers. So we already have one question here. It says, can your tissue dissociation solution be used for organoids?
Yes, we have tested different kinds of organoids and they have been used. So for example, we have tested it on spinal cord organoids, all sorts of kidney organoids. And yeah, depending on the different sample types, we are able to support you in a few ways. We can share protocols. Our handbooks have suggested protocols. Or of course, if you're sending it to service, we will help optimize that for you.
Thanks. And the second question is, do I need to purchase an instrument for single cell analysis?
No, so that's one of the advantages I mentioned for the cell loading and reagent loading. You can actually do it manually, by which I mean you don't need to, you just need a normal lab equipment available and a pipette that will help you load the cells and beads and reagents into our chip.
Yep. Yeah, for interest of time, we'll stop the Q&A here. And if you have more questions for Sujata, you can continue adding questions, or you can reach out to us by email. I also share the email in the chat box. So yeah, so let's move on to our second presentation. It will be given by Dr. Ma Weide from Cytech. He's an application scientist. He has worked extensively on immune cell profiling and multicolor flow cytometry. And let's invite Dr. Ma now to give his presentation. Dr. Ma, please.
Good morning, everyone. I'm excited to have this webinar to share this project where Cytech and Spectral collaborated to provide a solution for tissue flow cytometry. Today, we are talking about 20-color immunoanalysis of tumor infiltrating lymphocytes in lung cancer. If you are familiar with flow cytometry, you may know I'm talking about spectral cytometry when I mentioned 20 colors, because it's not easy to handle 20 colors in one tube for conventional cytometry. So what's the difference between a conventional cytometer and a spectral cytometer? We know that the emission of a fluorophore is a spectrum. A conventional flow cytometer detects the emission peak of a fluorophore. The number of detectable fluorophore is limited by the number of available filters. Spectral cytometer overcomes this limitation by employing a multi-channel detector. This detector can distinguish the shape of emission spectra along a large range of continuous wavelengths. Some fluorophore may have the same emission peak. They cannot be distinguished by conventional flow cytometry, but their spectrum may be different from each other, so they can be distinguished by spectral flow cytometry. By capturing the entire emission spectrum, spectral flow cytometry allows for better differentiation between overlapping fluorophores, enabling the use of a wide range of fluorochrome in a single tube. This diagram will show you my laser. This diagram here shows a 5-laser spectral flow cytometer. We can see each laser equipped with a multi-channel detector, as the final spectrum of this fluorophore is a combination of all the spectrum. We can see for BV750, the peak is at V14, but there's small peak at the UV14 and R6. We can use this small peak to distinguish different fluorophores. Spectral flow cytometry is revolutionizing cell analysis by providing researchers with a more nuanced understanding of cellular diversity and function. It has many applications. Here I list a reference about its application in cancer research, from biomarker discovery to vaccine and drug development to immune monitoring and cell functionality. You can find the original paper for details. For cancer therapy, we can target on tumor cells or non-tumor cells. Conventional radiotherapy and chemotherapy target on the tumor cells. They directly kill the tumor cells, but the residual tumor cells may survive. With the advancement in immunotherapy, highlights the critical role of non-tumor cells, including the immune cells, fibroblasts, and the extracellular matrix. These cells components compose the tumor microenvironment, TME. For TME study, can help to find new treatment strategies. A lot of different techniques have been used in TME characterization, like flow-based technique and imaging-based technique and omics technique, including single-cell sequencing and proteomics. For flow cytometry-based technique, we can analyze thousands of cells per second, and now using spectral cytometry, one can detect as many as 50 parameters at once. It is very helpful to deep immunophenotyping. What we did in this project was to obtain single cells from lung cancer samples and design a panel to characterize TME using spectral cytometry. So after the sample was sent to our lab, they were raised and cut into small pieces, and then put them into this dissociation system for enzyme digestion. And this part was provided by Syngron. The single cell was stained with antibodies and acquired on a 3 laser flow cytometer and analyzed by spectral flow software. Our panel includes a viability dye and 20 antibodies. You can see that fluorochrome were provided by Cytec. For this panel, we include CD45 and EpCAM to distinguish immune cells and tumor cells. We have markers for the major immune cell subset, like T cells, we have CD3, CD8, and CD4. We have B cell marker CD19 and CD123 for dendritic cells. We have 14 and 16 for monocyte and the neutrophil. We have 56 for NK cell. We also have PD-L1 and PD-1 to see the expression on tumor cells and T cells. You can see this, what I mentioned, it's a intermediate filament protein. Its role is mainly focused on intracellular structural property and contribution to enhanced malignancy of tumors by its involvement in epithelial to mesenchymal transition. In recent years, extracellular rule for what I mentioned have been proposed, and in this study, we keep this marker to demonstrate its extracellular expression. And that one thing I need to mention, for CD11C, this clone 3.9 need divalent cation to binding. So if you use EDT anticoagulated sample, you need to wash the sample before stain. Okay, that's the introduction part. Let's say experiment part. We know that the enzyme digestion may influence the macro expression. So we checked the expression on blood or tissue samples. We can see from this slide that all markers can be detected. We can see CCR7 mainly expressed on lymphocyte. CD11C was much brighter on monocyte. CD123 expressed on basophil or dendritic cell. Its side scatter was between the monocyte and the lymphocyte. CD14 on monocyte. CD16 was brighter on neutrophil and dimmer on lymphocyte. We use lung cancer samples for PD-L1, EpCAM, and what I mentioned. These 3 markers expressed on tumor cells. We can see the CD4 expressed highly on lymphocyte and dimmer on monocyte. Then we tested the panel using full blood samples. From the time gauge, my screen may be stuck. Oh, I will go. We can see the cell flow was very stable during the experiment. There's no air bubble or clog. Then we use scatter area versus height to removing doublet. Using CD45 versus side scatter, we can get leukocyte and some subset like lymphocyte, monocyte, and neutrophil. From leukocytes, we can gauge the basophil, which is CD123 positive and DR negative. And for monocyte, can be divided to classic monocyte and non-classic monocyte based on their CD14 and 16 expression. For the lymphocyte, we can gauge T cells by CD3 and B cells by CD19. For T cells include NKT-like cells and CD4, CD8 positive cells. For CD4 and CD8 positive cells, we can see different memory subset and naive T cells based on their BCR7 and the CD45RA expression. We also see PD1 expression on the T cells. For CD3 and CD19 double negative cells, we use CD56 and the DR to separate the NK cells and the dendritic cells. Based on the origin of cells, lung cancer can be divided into different subtypes, including small cell lung cancer and non-small cell lung cancer. And the non-small cell lung cancer can be divided into adenocarcinoma and squamous cell carcinoma. We collected 15 samples, of which 12 with known classification. We have 9 males, and adenocarcinoma account for 9 cases. I will show you 2 flow data, one from adenocarcinoma and one from squamous cell carcinoma. This is flow data from lung adenocarcinoma sample. From the time gauge, the stability of the cell flow was very good. We used side scatter, 2 side scatter. Side scatter one from blue and side scatter from violet to remove the doublet and the debris. Then we gated on the live cells and separated them into tissue cells and immune cells. For the tissue cells, we will keep an eye on the expression of EpCAM, 1-mention, and PD-L1. We can see from this graph that PD-L1 mainly expressed on EpCAM positive cells. And for immune cells, we can gauge the myeloid cells, the basophil, the monocyte, and the neutrophil. And for the lymphocyte, we can gauge the T cells. The T cells include CD56 positive NKT-like cells and the CD4 and the CD6 positive cells. We can see PD1 expression on T cells, and using CCR7 and the CD45RO naive and different memory cells were distinguished. CD4 positive cells also include CD25 positive, CD127 dim or negative population, which we thought mainly were Treg cells. CD3 negative cells include B cells, dendritic cells, and NK cells. And for dendritic cells, were distinguished as myeloid DC and PDC. NK cells were separated into earlier NK, mature NK, and terminal NK. This is a squamous cell carcinoma sample. Just like the previous sample, the cell clustering is very clear. We can see the PD-L1 mainly expressed on EpCAM positive cells. This sample have less basophil, but all other populations remained. We can see the monocyte, the neutrophil, and the different T cells, and the B cells, and the different dendritic cells, and the NK cells. Okay, that's the data we have. The following slide will present data analysis part. We know based on the immune cell infiltration, tumor is distinguished as cold tumor and hot tumor. Cold tumor have less immune cell infiltration and had poor response to immunotherapy. From our result, we can say sample 11 and sample 13 have less immune cells. These two samples may be cold tumor. We also listed the immune cell subset, and from the right graph, we can see the majority cells are T cells in lung cancer. We further analyzed the T cell population. We know the acquisition of antigen experience is marked by the generation and the persistence of memory cells, which provide long life protection against the pathogen. From our result, we can say that the majority part is effective memory T cells. PD-1 and PD-L1 inhibitors are the most widely used immunotherapy type to date. PD-L1 expressed on tumor cells bind to PD-1 receptor on activated T cells, which leads to the inhibition of the cytotoxic T cell. Blocking the PD-L1 and PD-1 signal by antibody reactivates the exhausted immune cells in the TME and may eliminate the cancer cell. And the prescription of some inhibitors may need the confirmed PD-L1 expression. So we counted the positive rate of PD-L1 and PD-1 in each sample, and using 5% and 50% as the cutoff of positive and high expression. We can say from our result, 7 samples express PD-L1 and 5 highly express. We also say that PD-L1 and PD-1 have different expression patterns. Some samples have high PD-L1 but not PD-1, vice versa. This panel includes 20 markers, so it is difficult to find all the information with the two-dimensional plot. We know that dimensionality reduction can help visualizing high-dimensional data. So we take one sample to illustrate. From the left graph, the cluster represents the t-SNE result, and the color represents the manual gating result. We can say the result of the t-SNE analysis were generally consistent with those obtained from manual gating. However, t-SNE results offered additional insight, such as the identification of 3 distinct clusters within the CD8 positive cells. From the right graph, we can see what's the difference between the 3 clusters. We can see from the CD8 expression, all 3 clusters highly express CD8. But from the CD127 expression, we can say that one population express CD127. The other 2 didn't express. From the left graph, we can also see that some NKT-like cells and some Treg-like cells merge together. We don't know it's meaningful. If you are interested in one population, you can get on that gate and do t-SNE analysis again. Generally speaking, we described a method to isolate single cells from lung cancer tissue, and we have an immunophenotyping panel to see the major cell type in TME. Thank you for listening.
Thanks so much, Dr. Ma. So again, for any questions, please use the Q&A function. So we already have one question from Huang Wang. The question says, are all reference controls from healthy subjects? Did you experience the unmixing profile that's different in healthy and patient samples. So if you use healthy cells as your reference controls, will there be the unmixing is correct for healthy samples, but will that also be correct for patient samples?
We use healthy samples and patient samples and the bead as a reference control. You can compare the spectrum between the different reference control. If they are the same, the similarity index about 0.99, then you can change each other.
Yep. Thank you, Dr. Mao. Is there any further? There's one question. How many cells do you require per sample? And was the tissue dissociation fully automatic?
For cell population like T cells, I think 10,000 cells may be enough. But for rare cell population like dendritic cells or basophils, hundreds of thousands of cells were recommended.
The second part of the question was the tissue dissociation automatic?
Sorry. Yeah. The second question, when sample sent into the lab, I need to cut into small pieces, then put the small pieces into this into the dissociation system, and for enzyme digestion. Yeah.
Thank you. The next question is, what's the maximum number of colors you would recommend in a single experiment?
From published literature, we can detect 25 colors for 3 laser for spectral flow cytometry, and for a 5 laser cytometer, we can handle 50 colors. But if need experience for beginner, I suggest you can refer the published literature.
Yep. The next question is, can the same flow cytometry panel be used for sorting?
Yes, we have 4 spectrum sorters, and all experiments that are designed in analysis cytometry can be transferred into sorter.
Yeah, great. Yeah, previously there was also a question for Dr. Sujata on tissue dissociation. So how fast would the automatic dissociation be compared to manual? And the answer is the automatic dissociation is about 15 to 30 minutes, depends on tissue type. Yeah. Is there any further questions from the audience? If not, we're approaching the end of the webinar. Thank you very much to both our speakers today. And we would especially like to thank Cytech for jointly organizing today's event. So everyone, please share your feedback through the survey after the webinar. And thanks again. See you next time. Bye-bye.